Good afternoon, everyone. For our group's research project, we were assigned to identify various virus-causing cancers. Each member of our group chose a virus to highlight and share critical information based on research conducted throughout the semester.
Hello, guys. My name is Alexander Nguyen, and I'm here to talk to you guys about the hepatitis V virus. What is HPV? The hepatitis B virus, or HPV, is a viral disease first found in 1965, and it targets the liver. The virus acts to cause inflammation and infections in the liver. The inflammation causes a perpetual cycle of degradation and repairing of the liver. This can result in liver failure, liver cancer, and cirrhosis, which is permanent scarring of the tissue. The disease causes inflammation in the liver through infections. HPV can either be acute or chronic. Acute HPV usually lasts to at most 6 months, while chronic typically lasts the whole lifetime. The severity of HPV is dependent on the age of the patient, as the CDC has noticed that 90% of infants develop chronic HPV compared to 2 to 6% for adults. It is also important to treat HPV as soon as possible before it can develop into chronic HPV. Risk factors and symptoms of HPV. Due to HPV being an infectious disease, everyone is at risk for HPV. It gets contracted through bodily fluids, saliva, or semen from someone who is infected by the virus. It is very important to keep any open wounds covered and sanitized, keep away from unprotected sex, and unsanitary needles. The symptoms to look out for include jaundice, which is the yellowing of the skin and eyes, dark urine, fevers, fatigue, abdominal pain, nausea or vomiting, loss of appetite, joint pain, and gray stools or excretion. Now, what makes HPV very dangerous is the fact that it can be asymptomatic, meaning most people who are infected with the virus never show any symptoms. Although it is important to seek out any medical attention if you notice any of these symptoms, typically 90 days after getting infected, because left neglected, the virus will continue to attack the liver and eventually change the acute HPV into chronic HPV. How does HPV cause cancer? A lot is still unknown about HPV, like how it specifically causes cancer, but HPV has been linked to be the main perpetrator of 60% of liver cancers in the world. The inflammation caused by HPV wreaks havoc on the liver by changing the DNA of liver cells and interfering with signal pathways. This can cause mutations in the cells like unnatural and uncontrolled reproduction of cells, and thus creating a tumor causing liver cancer. Now, as mentioned before, HPV can cause cirrhosis, which is permanent scarring of liver tissue. The scarred tissue acts as a plug and does not do any of the functions that the healthy liver tissue used to do. This leaves the liver vulnerable and more volatile to becoming cancerous cells. Keeping HPV under control. There is no current cure for HPV. For people with acute HPV, a healthy lifestyle will help clear the virus away, while those with chronic HPV might need to take medicine to treat the symptoms, but it does not cure the disease, and they come with a variety of side effects. However, a vaccine has been created and will grant immunity to the virus. As mentioned earlier, it is important to have children vaccinated as soon as possible to prevent early chronic HPV. Finally, since HPV can be asymptomatic, the CDC advises everyone to get screened at least once as an adult. As it stands currently, HPV is still a huge issue today, as 292 people are diagnosed with the hepatitis B virus.
Hello, my name is Jeremy Deneville, and the virus I will be presenting today is HIV, or better known as the human immunodeficiency virus. What is HIV? The human immunodeficiency virus, or HIV, is a virus that causes the overall weakening of the immune system by attacking and destroying the cells responsible for fighting off infections, like CD4 white blood cells. HIV doesn't kill, but with the weakening of the immune system, HIV makes it easier for opportunistic infections to cause serious illness or death. And obviously, as we all know, probably, or most likely, HIV can be spread by multiple avenues, such as, for example, blood transfusions, organ transplants, etc., etc. How does HIV function inside the human body? This is an old study, but it's still applicable to what we're going to talk about. So basically, HIV consists itself from detection by hijacking the CD4 lymphocytes, more commonly known as the helper T cells, which is also known as the HIV reservoir. And we have this more recent study which help us better, understand how HIV functions. And it basically the invading virus utilizes the reverse transcriptase enzyme of the helper T cell to convert its viral RNA to viral DNA. And once it completes that, the viral DNA will insert itself into the genome of the infected helper T cell, resulting in the new HIV copies from the influx of HIV proteins in genetic material. From here, HIV copies will go on to infect other helper T cells and start to cycle over again, resulting in increased HIV levels. So once the immune system realizes that it has been invaded by a viral infection, which is being HIV, it goes into like a response mode. In this response mode, it first starts off by, having the infected helper T cells release restriction enzymes called restriction factors, which are antiviral proteins to stop the virus production occurring inside the cell and prevent further virus from leaving the cell. Outside the cell, the immune system responds by having B cells, white blood cells produce and attach neutralizing antibodies on the surface of the virus to prevent them from hijacking other healthy helper T cells. The last response during this phase from the immune system comes from the activation of the killer T and natural killer cells, which go out and, kill like the HIV infections that, are just like kind of flowing in the body or they come across. And they do this by, puncturing basically the membrane of the cells, and then, it goes into self-destruction. All these responses causes HIV levels to drop, resulting in a stop to the immune response. HIV counterattack. All we really need to know for this slide is that HIV basically undoes all the work that the immune system has just done. It does this by basically deactivating the antiviral proteins inside the cell to continue, virus production and undergoing multiple mutations to avoid again getting detected by the immune system. And as it continues, the virus slowly weakens the immune system and stops responses from occurring while also increasing the levels of HIV again inside the body. Due to the strain on the immune system, it causes the virus to become unable to produce any more helper T cells and results in the development with enough time of AIDS. HIV contributes to cancer development by making it easier for opportunistic infections to infect an individual, thus increasing the risk of developing cancers because of the compromising of the immune system caused by, HIV. And with a weak immune system, this could let newly developing cancer cells live long enough to grow and replicate because the immune system is too weak to destroy those cells that are, becoming abnormal and developing into cancer cells. And it has been shown by multiple studies that infections of HIV or AIDS has been linked to increased development of multiple cancers, as I listed at the bottom of this slide.
Today I will be talking about human T lymphotropic virus 1, also known as HTLV1. HTLV1 is an oncogenic human retrovirus, so instead of using DNA, it uses RNA like other retroviruses. And it can cause a type of cancer called adult T-cell leukemia or lymphoma, depending on which one it affects. Adult T-cell leukemia lymphoma is mostly found in southern Japan, the Caribbean, Central Africa, and some parts of South America, which means that the HTLV1 infections are rare in the United States. And it is similar to HIV, but it does not cause AIDS. Also see in the image that the HTLV leukemia cells are like colored and like they look like flowers compared to other normal cells. HTLV1 is a distant relative of HIV1 as they have similar structures with different proteins, obviously, and In HIV-1, it induces apoptosis of CD4 lymphocytes, which means that it kills the cells, while in HTLV-1, it transforms and induces uncontrolled proliferation of T cells. So it pretty much makes the T cells divide and grow rapidly and uncontrollably, unlike HIV. And ATL cancer happens when the HTLV-1 virus would attack and stimulate the infected T cells, which should cause them to grow via constant exposure to Tax and HPZ proteins. We can see in the image that in HIV-1 and HTLV-1, the matrix is different, where HIV-1 will contain P17 proteins and HTLV-1 will contain P19 proteins in the matrix. And also in the nucleocapsid, HIV-1 will contain P7 proteins and HTLV-1 will contain P15 proteins. The HTLV-1 virus can also spread the same way as HIV, such as through exchange of bodily fluids. Unprotected sex and needle sharing are major ones. Infected mothers can also pass the virus down to their children through breastfeeding. So to reduce the risk of infection, an infected mother should not breastfeed their child. The HTLV-1 virus infects the unaffected T cells, mainly through cell-to-cell contact. It has the ability of attachment and fusion to the cells with the help of HSPGs, glucose transporter, GLUT1, and neuropilin-1. There are 3 possible entries also, which is transmigration of infected macrophages, transcytosis of viral particles, and release of virions. We can see in the image above the 3 possible mechanisms for viral entry, which is the first one being transmigration of the HTLV infected macrophages, the second one being the transcytosis of viral particles, and the third one being the HTLV-1 virions moving through with a damaged mucosal. All 3 of them are from the basal surface. Most people with an HTLV-1 infection are asymptomatic and do not develop conditions or symptoms that can be linked to the infection. And when diagnosed, there are 4 clinical subtypes: acute, lymphomatous, chronic, and smoldering, with the more aggressive subtypes being acute and lymphomatous. Since most people infected are asymptomatic, not a lot of research has been done on HTLV-1 as compared to HIV-1, which is more dangerous. HTLV-1 can also cause other diseases that are not ATL cancer, such as myelopathy or tropical spastic paraparesis, which is a chronic inflammatory disease of the central nervous system, which can cause weakness in the lower limbs, back pain, and bowel and bladder dysfunction.
Hi, this is Alyssa Raymondville, and I'll be presenting to you today about the Epstein-Barr virus. The Epstein-Barr virus is a gamma herpes virus, meaning that it reproduces at a more variable rate than other viruses within the families of Herpesviridae. EBV can grow in vitro settings and in vivo settings. Some of the ways that someone can contract EBV is through respiratory secretions such as mucus. EBV works to attack the B lymphocytes of the host along with other cells that are immunocompromised. Therefore, people who receive organ transplants or stem cell transplants are at high risk. The symptoms of EBV can vary depending on the person's age and immune system. Specifically, young adults aging from 15 to 24 years old may experience pharyngitis, adenopathy, and fatigue. Adenopathy involves the swelling of the lymph nodes, and lymph nodes are present all over our bodies from our heads to our toes. So, for example, when you go to the doctor for an exam, they may feel around your neck to see if there is any swelling present to detect sinus infections. EBV has been discovered to be related to the cancer Burkitt lymphoma. Burkitt lymphoma was the first human tumor to be associated with the virus and is the most common childhood cancer in areas where malaria is holoendemic. BL is endemic in Africa and can lead to death if not treated promptly. And so an endemic disease is one that occurs within an area, whereas a holoendemic disease is one that has a high prevalent level of infection during the early stages of life. Some of the ways BL is diagnosed is through full blood count measures, electrolyte measurements, and liver function tests. As for low-developed countries, diagnostic measures include cytological examinations with the fine needle. EBV has relations to other diseases such as Hodgkin's disease, nasopharyngeal carcinoma, and post-transplant lymphoproliferative disease. Hodgkin's and nasopharyngeal relation to EBV was discovered by the presence of EBV DNA in Hodgkin cells and tissues in serological studies of undifferentiated nasopharyngeal carcinoma cells. PTLD describes the lymphoproliferations that occur in the B cells after impaired immune systems receive transplants, which can lead to B lymphocytes being taken over. PTLD can occur with any organ transplant within the body, such as the lungs, kidney, and heart. A common one includes swelling of the neck, armpits, or the groin. Treatments for EBV include immunotherapy, antibodies, and antivirals. Immunotherapy helps to use the person's own immune system to fight the cancer, which can lead to immune system boost or changes. Antibodies such as rituximab helps to destroy cells that have receptors for EBV DNA, which is present on all mature B cells, and these receptors are called CD21. Antivirals such as acyclovir and ganciclovir help the body fight off the virus and relax the symptoms and shorten the duration of the virus presence in the body. Also, sinus infection remedies such as resting and drinking lots of fluids help to relieve the symptoms of EBV, but does not exactly cure it, unfortunately.
The Kaposi sarcoma-associated herpesvirus. What is KSHV, or the human herpes virus 8, HHV8? Well, it's a gamma herpes virus which causes infectious mononucleosis. It is also in the same family as the Epstein-Barr virus. Epithelial cells are infected by KSHV, which brings genes into cells and causes rapid and continuous division and extends the lifespan. Endothelial cells may also form new blood vessels, increasing production of inflammation, causing chemicals which create cancer cells. The causes of Kaposi sarcomas are triggered by compromised immune systems and especially common in patients with HIV. KSHV can also cause multicentric Castleman disease, primary fusion lymphoma, or other KSHV-associated inflammatory cytokine syndromes. Pathobiology of KSHV. After infection, the genome is maintained in the episome of the host cell nucleus. KSHV can infect a wide variety of cells. After infection, latency is established where only a few genes are expressed. These genes tend to stay in the latency locus, where you can see the ORFK12 and caposins, latency-associated nuclear antigens, and many others, which help transcript and promote the growth of the proliferative signals and help the survival of the infected cells. Physiological signals can result in the virus entering into the lytic phase, where there's no direct role in endothelial growth and the cell dies. Genes evade innate and specific immunity, which induces proliferation and prevents apoptosis, which contributes to the oncogenesis and angiogenesis. KSHV proteins promote activation of motor pathways and the upregulation of interleukin. It rarely leads to KS, but other cofactors such as immunosuppression are required for the virus to induce a tumor. Types of Kaposi sarcomas. There are 4 main types of Kaposi sarcomas. The first one is the epidemic or AIDS-related Kaposi sarcoma, which is traditionally transferred via saliva transmission, particularly in homosexual males. Then there's the classic or Mediterranean Kaposi sarcoma. So older men of Mediterranean, Middle Eastern, and European descent traditionally get this in their childhood through mother-child saliva. After they've had the virus, cancer may occur later due to age-related immunity decline. The third one is the endemic or African Kaposi sarcoma. This is due to a high percentage of KSHV presence in the population. It is also transferred through mother-to-child saliva, and it's often seen in younger boys. The final one is transplant-related Kaposi sarcoma. Immunosuppressant drugs are taken to undergo organ transplants, which compromise the immune system. If organs are already affected by KSHV, there's a higher chance of getting it, especially in countries where KSHV is more common. Symptoms, diagnoses, and treatment. Symptoms are seen through purple patches or nodules in the skin or mucous membranes, which can spread to the lungs. These nodules often disfigure your feet, legs, and face. On the mucous membranes, you may find similar disfigurations in the mouth, anus, or anywhere on the GI tract. Inside the body, you may also find it on the lungs, which can cause internal bleeding, trouble breathing, and anemia. Lymph nodes are also common, particularly in the groin, and painful swelling in the legs. Diagnosis is very rare, but when it is found, it's through skin biopsies, chest x-rays, bronchoscopy, or endoscopy. Now, this is incurable, but treatable through improving immunity function, through using immunotherapy or other therapies such as chemo and local ones. The mortality associated with this virus is usually through HIV or AIDS-related diseases.
Today, I will be talking about Merkel cell polyomavirus. Polyomaviruses are a family of naked or non-enveloped circular DNA viruses that can infect a wide array of mainly mammalian hosts like humans and monkeys. They display icosahedral symmetry of the nucleocapsid. Merkel cell polyomavirus is one of 7 known human oncoviruses and causative agent of Merkel cell carcinoma. Merkel cell carcinoma is a rare and metastatic type of skin cancer of the Merkel cells that are present in the basal layer of the epidermis, directly above the basement membrane. These cells are responsible for the sensations of light touch and are a type of neuroendocrine cells. Aside from Merkel cell polyomavirus, Merkel cell carcinoma can be caused by excessive sun or UV exposure and aging. Merkel cell polyomavirus was very recently discovered in 2008 during a study of Merkel cell carcinoma, a type of human skin cancer that commonly occurs in individuals with weakened immune systems. Scientists discovered the presence of the viral genome through digital transcriptome subtraction, a type of differential analysis in which they systematically removed all known human sequences from the host cell's DNA to identify any foreign or non-human DNA. They discovered Merkel cell polyomavirus sequences within many of the Merkel cell carcinoma tumor cells, which suggested that infection by the virus predated the formation of the tumors, leading them to believe that it contributed to the development and is a causative agent of Merkel cell carcinoma. The mechanism of transmission of Merkel cell polyomavirus is unknown. However, the intact virus particles have been found shed by healthy skin cells, they're present in respiratory secretions, and are present in gastrointestinal tract samples, though it is the generally accepted idea that the skin is the major reservoir for this virus. This virus is highly prevalent in the human population, with infection usually occurring in childhood and adolescence and is asymptomatic. By the time they reach adulthood, 90% of individuals in the U.S. are positive for Merkel cell polyomavirus antibodies, specifically antibodies for viral capsid protein 1, which makes up a majority of the pentamers that encase the viral genome by way of the capsid. However, despite how commonly it is encountered, only around 1 in 130,000 people develop Merkel cell carcinoma, and this typically only occurs in the elderly, aged over 50 years, or in immunocompromised individuals, such as those with HIV. Common symptoms of Merkel cell carcinoma are red or purple fast-growing tumors on the skin, usually on the face or arms, as well as swollen lymph nodes. The mechanism of infection and integration of the viral genome into the host genome can be simplified and put into 4 steps. The first step, attachment. In order for infection to occur, Merkel cell polyomavirus must first bind its viral capsid protein 1 to sulfated glycosaminoglycans and then through a secondary interaction between VCP1 and the sialylated glycan receptors on the surface of the cell. Next, the virus is able to enter the cell via lipid raft-mediated endocytosis and is brought into the cell via endocytic vesicles, which actually provide the naked, non-envelope virus with a temporary viral envelope made from the host cell's plasma membrane. The envelope virus is taken to the endoplasmic reticulum, where the envelope and capsid are removed and the viral genome is brought to the cytosol. Merkel cell polyomavirus is unlike other types of viruses because the integration of the viral genome into the host genome is accidental and occurs randomly through double-stranded breaks in the virus genome and then integration into the host genome through non-homologous end joining, as seen here on this diagram as linear integration pattern. And for our last step, the transformation of the Merkel cells, it's important to note that the virus itself is not what causes Merkel cell carcinoma to appear and initiate tumor growth. The virally encoded T antigen expression acts as tumor promoters on the pre-existing Merkel cell carcinoma promoter cells.
Human papillomavirus, also abbreviated as HPV, is another example of virus causing cancers. HPV is a viral infection commonly spread through contact, causing papillomas or warts. While many types of HPV are generally harmless, at least a dozen are known to cause cancer. Most people infected with HPV are unaware because they show no symptoms. Low-risk HPV types may cause genital warts, but high-risk types can lead to various cancers such as cervical, vaginal, vulvar, penile, anal, and oropharyngeal cancers. It's important to note that most sexually active individuals will be infected with one or more types of HPV at some point in their lives. Cervical cancer is the fourth most common cancer in women globally, with the highest incidence and mortality rates in low and middle income countries. The primary cause of cervical cancer is persistent infection with high-risk types of HPV. Women with weakened immune systems, like those with untreated HIV, are at a significantly higher risk, being 6 times more likely to develop cervical cancer compared to women without HIV. Persistent HPV infection in the cervix, if left untreated, is responsible for about 95% of cervical cancers. It typically takes 15 to 20 years for cervical cancers to become cancerous, but in women with weakened immune systems, this process can be accelerated to just 5 to 10 years. Various risk factors influence cancer progression, including the type of HPV, immune status, the presence of other sexually transmitted infections, number of births, age at first pregnancy, hormonal contraceptive use, and smoking. HPV is also responsible for several other cancers. Anal cancer, for instance, is primarily caused by HPV, accounting for about 90% of its cases. Symptoms of anal cancer can include changes in bowel habits, blood in the stool, and anal pain. Oropharyngeal cancers, which affect the back of the throat, including the base of the tongue and tonsils, are also linked to HPV, causing around 60 to 70% of these cancers. Symptoms may include a persistent sore throat, ear aches, hoarseness, swollen lymph nodes, and unexplained weight loss. HPV is a risk factor for penile cancer, with studies estimating that 30 to 60% of cases are caused by the virus. Vaginal cancer, while less common, is primarily caused by HPV, particularly in women between the ages of 30 and 60. Lastly, HPV is a significant cause of vulvar cancer, accounting for about 70% of cases. This is especially concerning because genital warts caused by HPV are linked to an increased risk of vulvar cancer. Vaccination against HPV is important in preventing these cancers. Preteens, both girls and boys, should receive the HPV vaccine to protect against future HPV infections. There are 3 FDA-approved HPV vaccines: the 9-valent HPV vaccine, the quadrivalent HPV vaccine, and the bivalent HPV vaccine. All these vaccines protect against HPV type 16 and 18, which are responsible for the majority of HPV-related cancers. The HPV vaccine has proven to be highly effective. Since its introduction in 2006, there has been a significant reduction in HPV infections and related diseases. The vaccine is recommended at ages 11 to 12, and 2 doses are usually sufficient if the first dose is given before the 15th birthday. For those aged 9 to 26 with weakened immune systems, 3 doses are recommended. Most insurance plans cover the HPV vaccine, and it is widely available at doctors' offices, health clinics, and community health centers.
Hello, my name is Jacob Camp, and I will be going over the cancer-causing virus hepatitis C, also known as HCV. You may be asking yourself, what is hepatitis C? Hepatitis C is a viral infection that spreads through contact with infected blood. This transmission can occur through various means, such as sharing needles, receiving contaminated blood transfusions, or through other forms of direct blood-to-blood contact. The term hepatitis describes inflammation of the liver, a critical organ responsible for filtering toxins from the blood, aiding in digestion, and performing many other essential functions. When the liver is inflamed, its ability to function properly can be severely compromised. There are 2 primary forms of hepatitis C, acute and chronic. Acute hepatitis C is a short-term illness that occurs within the first 6 months of the exposure to the virus. In some cases, acute hepatitis C resolves on its own. However, if the virus persists in the body for more than 6 months, the infection becomes chronic. The chronic hepatitis C is a long-term condition that can last a lifetime and often leads to more serious liver problems such as cirrhosis, liver failure, and liver cancer. The hepatitis C virus has 7 genotypes, with a total of 67 subtypes. The genotypes are numbered 1 through 7. In the United States, genotype 1 is the most prevalent, accounting for the majority of HCV infections. Symptoms associated with HCV. Hepatitis C virus is commonly diagnosed through a blood test, which is essential for detecting the presence of the virus and determining the appropriate treatment. Treatment for acute hepatitis C is highly effective, with a 90% success rate. Most individuals with HCV do not exhibit symptoms initially, but both during the acute and chronic phases, they may experience fever, muscle aches, fatigue, belly pain, jaundice, joint pain, and even dark-colored urine. Jaundice is a condition that causes the yellowing of the skin and eyes as pictured on the left. Chronic HCV has also been linked to more severe health issues, including type 2 diabetes, lymphoma, lung cancer, and various other neurological disorders. This highlights the importance of early detection and treatment to mitigate the long-term health impacts associated with the hepatitis C virus. The link between HCV and cancer. In the United States, hepatitis C virus is responsible for approximately 50% of all cases of liver cancer among patients. Chronic HCV infection often progresses to cirrhosis, a condition where liver tissue is replaced by scar tissue, which significantly increases the risk of developing hepatocellular carcinoma and potentially impacting other organs such as the lungs. HCV primarily targets hepatocytes, the main functional cells of the liver, causing them to replicate the virus, leading to a widespread liver damage. This damage results in the accumulation of scar tissue, impairing liver function over time. Overall, HCV's impact extends beyond hepatocytes, disrupting other vital components of the immune system within the liver, including lymphocytes, Kupffer cells, and endothelial cells lining blood vessels. HCV treatment and prevention. When it comes to acute HCV, the most common form of treatment is antiviral medication, usually consisting of an 8 to 12-week regimen. In addition, for chronic HCV, antiviral medication is also prescribed. However, depending on the impact the virus has on the liver, a liver transplant may be necessary. Having a liver transplant does not cure someone of hepatitis C. The patient will need to continue taking antiviral medication for the rest of their life. Some simple ways an individual can prevent themselves from obtaining HCV is obviously no blood-to-blood contact, practice safe sex, prioritize washing and disinfecting your hands, avoid using illegal drugs, and stay away from dirty tattoo and piercing shops. Unfortunately, there is no current vaccine for hepatitis C, but as science continues to evolve, there is still hope. Another form of prevention doctors recommend when it comes to HCV is getting vaccinated for both hepatitis A and B viruses. Thank you so much for listening to our group's research presentation on virus-causing cancers. The following are the references used during our research. Are there any questions?